it's my honor to introduce Dr. Nicolas. Is a full-time professor of pharmacology at the University of Angers, specializing in molecular pharmacology, nanomedicine, and translational therapeutics. He obtained his PharmD, biochemistry and molecular biology, PhD in physiology and pathophysiology, and habilitation to direct research. His postdoctoral and academic career spans prestigious INSOM and CNRS research units, with progressive roles from temporary research associate to full professor since 2022. Dr. Nicolas' research focuses on endothelial plasticity, tumor microenvironments, nanomedicine, and cardiovascular pharmacology, with over 1,985 citations and an H index of 20. He has authored more than 35 high-impact publications, contributed to 2 patents, and serves on editorial boards of leading pharmacology journals. He has successfully supervised multiple PhD and master students and secured substantial research funding, including ANR, IRSPR, and European Co-Fund Breathe. In education, he leads master's courses in nanomedicine, cellular interactions, and therapeutics, and has authored foundational pharmacology textbooks. Administratively, he directs the Angers Postdoctoral Center, participates in national scientific councils, and contributes to regulatory advisory boards such as ANSM and ANSES. They are French regulatory authorities. He is also an active member of scientific societies, including the French Society of Pharmacology and Therapeutics and the European Association for Cancer Research, demonstrating leadership in both research and academic governance. With this introduction, I request Dr. Nicolas Clere to deliver his lecture. Thank you, sir.
Thank you very much. Thank you very much for your invitation. Today, I would like to talk about the endothelial cells differentiation to better understand its role in tumorigenesis and in drug resistance. To introduce my presentation, I have chosen to draw inspiration on part of an editorial written by Montserrat Rojo de la Vega, an editorial member of Cancer Cells. She wrote her editorial on 2023, and in its editorial, Montserrat described an holistic view of cancer. She described the role of cancer cells in an environment composed by different cells. She wrote that tumor cells do not exist in a void. The tumor microenvironment is a complex yet highly structured ecosystem. She concluded her editorial by a very interesting sentence. She concluded by this sentence: therefore, to understand the one, we must understand the other and recognize their interconnectedness. The tumor microenvironment has been described by Douglas Hanahan in different reviews. Douglas Hanahan described the hallmarks of cancer, and Douglas Hanahan described the hallmark of cancer by identifying cancer cells, cancer stem cells that can be differentiated, and these cells are in an environment composed by cancer-associated fibroblasts that are involved in tumorigenesis or in drug resistance, endothelial cells and pericyte that compose the vascular structure, and immune cells such as macrophages, lymphocytes, or dendritic cells. From disease discoveries, different molecules have been commercialized, such as immunotherapies or targeted therapies, such as tyrosine kinase inhibitors. While these molecules are very good to treat the cancer, different resistance have been discovered. The origin of disease resistance have been described in 2024 in a paper published by Rozowska. Different origins have been identified. The first origin is the tumor heterogeneity. In fact, cells during the tumor development, cells acquire genetic alterations that generate genetic diversity. The second hypothesis is correlated with the first, because if the tumor grows low, the tumor cells can acquire genomic alteration and then can acquire drug resistance. Tumor cells can also acquire genomic drivers, undruggable genomic drivers that can induce drug resistance. Among these genomic drivers, we can cite MYC or TP53 that are known to be involved in cell cycle. Another hypothesis is the selective therapeutic pressure exerted by radio or chemotherapy that can induce drug resistance. And finally, drug resistance can be explained by the cells that compose the tumor microenvironment. Among these cells, endothelial cells play a key role in drug resistance. Endothelial cells are at the crossroad of different pathophysiological processes. In fact, in tumorigenesis, endothelial cells can be involved in angiogenesis. Angiogenesis has been discovered by Julia Folkman in 1971, and different other works have demonstrated the role of angiogenesis. Angiogenesis is defined by the formation of new vessels from pre-existing. Angiogenesis is very important to explain the metastatic process, for example, and angiogenesis from the data from Folkman and Peter Carmelit, different molecules have been commercialized to target VEGF or its receptor and to treat different solid cancer. The second process is vascular mimicry. In vascular mimicry, endothelial cells are indirectly involved in this process because vascular mimicry is an epithelial cell differentiation in an endothelial compartment, an endothelial environment, that is, in an environment in which angiogenic factors are mainly expressed. And the third mechanism is endothelial to mesenchymal transition that first discovered in fibrosis, in lung fibrosis, and discussed in cancer. Endothelial to mesenchymal transition is endothelial differentiation induced by different mediators. During this process, endothelial cells lose their properties. For example, they lose their cell-cell junction or their polarities. Endothelial cells lose also their endothelial markers, such as CD31, VE-cadherin, or von Willebrand factors. These differentiated cells acquire new properties, such as mesenchymal properties, as an enhancement of migratory properties. These cells acquire also mesenchymal markers, such as N-cadherin, alpha-smooth muscle actin, SM22-alpha, vimentin, or others. In a recent review, we compare disease endothelial to mesenchymal transition to sprouting angiogenesis that favor the production of cancer-associated fibroblasts involving the extravasation and metastasis, so tumor growth. As previously said, different molecules induced endothelial to mesenchymal transition. TGF-beta, hypoxia, Wnt, chemo or radiotherapy. And we evaluated the role of oxidative stress. In fact, oxidative stress is mainly expressed in solid tumor. ROS and ADPH oxidase are mainly expressed in different solid tumors. And the first part of our project was to describe the role of oxidative stress in endothelial to mesenchymal transition. This work are conducted by Valentin Platel during his PhD, and Valentin has evaluated the ROS production from conditioned media called CM during this presentation by evaluating DCFDA fluorescence intensity by flow cytometry. Interestingly, we observed, after 48 hours and 72 hours, an increase of ROS production when the cells in cancer cells, and we evaluated the role of NOX1 and NOX2 by using 2 inhibitors, and interestingly, we observed after 24 hours, 48 hours, a decrease of ROS production, and a significant decrease of ROS production after 72 hours confirmed the role of these 2 enzymes in CM-induced ROS production. Then we evaluated the role of these 2 enzymes in tubulogenesis. Tubulogenesis is the capacity of UVEC to form capillaries on matrigels. Then we observed a significant decrease of capillary length when the UVEC cells were treated with CM. And we observed, interestingly, after 24, 48 and 72 hours, we observed that the co-treatment with inhibitors against NOX1 and NOX2 prevent the decrease of capillary length. So we confirm that inhibiting NOX1 and NOX2 prevent CM-induced endo-MT. We discovered 2 enzymes that are able to induce endo-MT. And then in the second part of this work, we evaluated the content of the CM by proteomic analysis. And we identified, Diane Le Chevalier, during her Master 2, identified a protein, spark protein, known to favor the tumorigenesis. We evaluated the role of spark in a clinical study, and we quantified the plasma level of spark in a cohort of 40 patients with stage 3-4 melanoma, and we confirm a correlation between the level, higher level of spark and the BRAF status. So we identify a correlation between BRAF mutation status and plasma spark concentration in melanoma, in metastatic melanoma. We then evaluated the endothelial to mesenchymal transition in non-small cell lung cancer. That is an aggressive cancer with a 5-year survival rate less than 20%. We evaluated the role of endothelial to mesenchymal transition in this cancer, and Clara Bourreau, during her PhD, has evaluated the role of endothelial to mesenchymal transition. First, by immunochemistry study, we showed that cells, UVEC endothelial cells treated by different CM from NSCLC, the treatment increase the actin stress fibers and decrease von Willebrand factors and endothelial markers. So this results suggest that CM induced morphological change in UVEC. Then we evaluated the migration of disease-differentiated cells, and we observed, interestingly, that UVEC cells treated by each CM are able to induce, to enhance their migratory properties. And interestingly, after 72 hours, we observe an enhancement similar that observed with TGF-beta and EL1, the positive control of our experiments. Then we discussed the phenotype of the treated cells by flow cytometry. And on this slide, I present only 2 CM, and we observe a significant decrease after 48 and 72 hours, a significant decrease of the double staining von Willebrand factors and alpha SMA, and a significant increase of cells that expressed alpha SMA. So this is results confirm a modification of the phenotype of endothelial cells, and then CM are able to induce the transition of treated UVEC. Finally, we identified the content of the CM, and we observed 5 clusters for each CM. And interestingly, we identify a correlation heat map with proteins that involved in matrix remodeling or epithelial to mesenchymal transition. And we identified osteopontine as a protein involved in endothelial to mesenchymal transition. And interestingly, we showed that osteopontine is correlated with less prognostic of lung cancer, and osteopontine is inversely correlated with CD31, an endothelial marker We realized all this experiment in 2D model. But a paper from Kim in 2019 has described the role of 3D model in endothelial to mesenchymal transition, and he showed that treatment of endothelial cells with medium from 3D model enhanced the endothelial to mesenchymal transition with a higher alpha SMA and a less CD31. So we designed different spheroids, and Eva Martin, during her PhD, designed different spheroids, monocellular spheroids, multicellular spheroids with UVEC or Schwann cells. And this slide shows live cells in green and died cells in red. And interestingly, we evaluated the endothelial to mesenchymal transition, and we showed that VEGFR, von Willebrand factor, or CD31 are significantly decreased in endothelial cells in the 3D model, and we observed a significant increase of SM22, vimentin, 2 mesenchymal markers of endothelial cells. So interestingly, we have shown that endothelial cells, endothelial to mesenchymal transition is an important process in tumorigenesis. In fact, there is different mediators that induced endothelial to mesenchymal transition, TGF-beta, Notch, oxidative stress, and different molecules decrease endothelial to mesenchymal transition, such as VEGF, miRNA, or different chemical inhibitors. Different molecules are able to decrease endothelial to mesenchymal transition. Imatinib, a tyrosine kinase inhibitor, hydrocortisone, or spironolactone. But all the experiments described by Mann in 2019 were realized in vitro assays. So it's very important to confirm these results in vivo or ex vivo models. Finally, endothelial to mesenchymal transition could be interesting to treat different diseases. In fact, endothelial is differentiated in mesenchymal cells, and then mesenchymal cells could be differentiated in different other cells. These other cells can be used, for example, in tissue engineering to treat different disease, and maybe these cells could be interesting to treat, for example, cancer. To conclude, I would like to thank Eva Martin, Trea. Trea is a PhD student from India who arrived in our lab at the beginning of October. Valentin Platel, Clara Bourreau, Diane Le Chevalier, Flavien Vessaguet from Mitovask lab, Professor Ludovic Martin from the Department of Dermatology, Dr. Lucas Treps and Dr. Isabelle Cor from the CRCI de Hénin lab in Nantes, in France. And to finish, on behalf of the French Pharmacological Society, in which I am part of the advisory board, I would like to express my sincere gratitude for the excellent welcome, my gratitude to the Indian Pharmacological Society, to Dr. Sandeep, and to all organizers of this meeting. It's a pleasure for me to be here. Thank you very much.
Thank you, Dr. Nicolas. And we need to keep up the time, so we will allow again one question to be asked to Dr. Nicolas Clere. Anybody, one question? Okay, if not there, thank you very much, Dr. Nicolas. It was wonderful, listening to you.
Thank you very much.